Okay, good morning. Welcome to the 2024 WAIS Workshop. I'm Matt Siegfried. I'm one of the organizing committee members. We have two more organizing committee members here. Knut Christensen, raising his hand over there. And then our local host, MacKie, just raised her hand. MacKie will come up and give a little intro about University of Florida in just a second. But I thought I would give a little intro to the WAIS Workshop to get things started. You'll also get a little bit of logistics from me at the end. But the thing I really wanted to get up here and talk about is that we're in a very, very exciting time for our community. We're ramping down the International WAIS Glacier collaboration. The last field season just happened. One more field season? No, we're done. We're done. And we need to think about what's next for the West Antarctic ice sheet community and how we study marine-based ice sheets as glaciologists and collaborators in other parts of the Earth system. We have the upcoming International Polar Year in 2032, 2033, and we have a NASA decadal survey coming up in 2027. And so this is our time to think big. And so because of that, kind of building on the history of 30 years of the WAIS Workshop, it is our responsibility as WAIS Workshop participants, the WAIS Workshop community, to communicate a clear plan for our community and for our science to launch all of these big projects. And so that's really our goal this week, is to get our act together and organize and think about what the big picture science we want to get done over the next 5 to 10 years is. We started this process last year in 2023 in Cloquet, Minnesota, I guess is where it was. Available at this QR code, along with a Google form where you can give us feedback on this. This will be, I haven't printed it yet, but because we just made it last night. But that QR code is going to be printed and posted in the back. So you can scan this. It'll also be on our webpage. This takes you to a Google form where you can look at this 3-page document of notes, give us feedback. We want to hear from you. We want your input. We really want your prioritization, right? Like, what should we be tackling? Cool. Here's what it looks like. Input for WAIS Workshop Community and Science Plan. And then you can scroll down. There's a free-form text box. Give us your thoughts, please. And then this is just the start. There's a little preamble to the document we wrote about how we came up with this 3-pager and where we're going with it. The goal is, by March, we have an actual white paper that's deliverable to whoever needs it to launch IPY. We already have a National Academy's IPY committee. There's going to be a workshop dedicated to that. This is going to flow into that workshop. We can hand it off to NASA as well. Cool. On that topic, the WAIS Organizing Committee, there are 6 of us. 3 are in the room. The other 3 are virtual on the live stream: Peter Neff, Brooke Medley, and Lauren Miller. We've been doing this for a while, and people are cycling on and off. And we wanted to make that the way we replace members of the organizing committee a bit more inclusive than the kind of white smoke coming out of the church type thing we've been doing recently. That was a joke. We don't actually do that. So if you are interested in taking a leadership role in this community, please let us know. We have yet another Google form. This is a different QR code with a different link. This is already on our homepage. It's been there for 2 months now, I think. Please nominate a peer or self-nominate someone you think that would be great at helping organize this community. We want fresh faces. We want people of all career stages, so please get on there and nominate. Some logistical announcements. The first one, because this seems to always be a problem at every conference I go to, we have 2 laser pointers for speakers. You get to choose your own adventure here. One is analog, right? Like you can see this on the wall over there. It can also go back and forth. One is digital. Look at this, right? No matter where I point, it comes up. It's supposed to be a laser pointer. Oh, there it is. Digital. The digital one lets the live stream people see where you're pointing. So it'll be saved for all eternity on YouTube. But it's a little funky to use. It's a digital laser pointer. It's not natural. So choose your own adventure here. Both work. We have wonderful student awards once again this year. There are QR codes plastered everywhere. Everyone in this room is a judge. Your feedback will be used to do these student awards. All the students who have posters, there's a little tag for you to leave on your poster that says, I'm a student, please give me feedback, with the QR code to go to the form to do awards. Leave that on your poster. If you're not a student, just take it off your poster. I put it on every poster board yesterday. All of these QR codes around the room will take you to the student award form. And so we are crowdsourcing these awards. It's 4 quick questions and then open feedback that we'll pass along to the students. Don't forget to do that. Marion, do you have your backpack? The students win a beautiful WAIS Workshop backpack. Oh, Roger, we have 2 winners in the room. They're great backpacks. So we can only give these away if people partake in the award judging. As I already mentioned, we have a live stream. All of the talks are going to be archived with permission of the speakers. We also have a Slido for remote questions. You don't have to be remote to use the Slido. Just go to slido.com or whatever their website is and say WAIS 2024. And you yourself in this room can give a digital question that someone will hopefully read out loud. And just so everyone knows, most of the community is not aware of this, but we've been archiving these talks since 2019. We now have almost 76 hours of WAIS Workshop science up online for anyone to view. This is really handy for classes, for those of us who teach. I've built it into curriculum. It's really great for bringing in new collaborators, right? Like, if you want to spin a collaborator up on the latest science coming out of our community, you don't need to sit around and wait for the next WAIS Workshop or AGU or whatever. You can just hand them one of the digital agendas from a WAIS Workshop. It has a link to every single talk directly. Use it. It's there for you. 241 individual presentations you can watch online. Yeah?
One of my co-authors is reluctant to have it posted because we have something
that's fine.
updated,
Yeah.
and so we can opt out.
Yeah. So every year, a couple people are like, hey, it's not published yet, or there's something wrong in it. We're happy to take them down, but for everyone else, they're up there. New this year, based on some feedback, all good on the live stream? Cool. Based on some feedback from the early career folks last year, we're gonna give any non-presenting new attendees the option to introduce themselves during the poster lightning talks. We've never done that before. So if you are not presenting and this is your first WAIS Workshop, how many people is this your first WAIS Workshop? Love it. How many people have been to 15 or more WAIS Workshops? A couple of us in the room. Reed was at the very first pre-WAIS Workshops, the C-Rise Workshop. What did we call it? Just C-Rise? C-Rise. So we have a long history here. If this is your first WAIS Workshop and you are not presenting, we want to know who you are. We want to know what you're interested in. So after all the posters, Allie is the MC for the posters. After those lightning talks, stand up or get in the line, introduce yourself, tell us who you are, where you're coming from, what you're interested in. Because we want to involve you in the community, we want to know who you are. Last thing, we have a code of conduct. It's posted behind Marissa, who's working the check-in desk. That's for the conveners. So you remember to yell at people if they violate the code of conduct. My phone number and Mickey's phone number are both on the code of conduct posted back there. If anything comes up 24 hours a day, give us a call, give us a text. All of this boils down to be a good person. You know, we want this to be bullying and harassment free. This is a very tight-knit community. That's kind of the whole idea. And so please take this seriously. But I do want to mention that we want to take the code of conduct seriously. That doesn't mean we shouldn't be disagreeing about science. We can still do that professionally, respectfully, and scientifically, right? This is what we're here to do. We need to argue about our science, debate about our science, to get the best science, to launch the best projects. And so please don't be afraid that just disagreeing about science is a violation of code of conduct. It is not. I want to make sure everyone knows that. We should be thinking about holes and plugging them in our science. Any questions about that? I feel that's very important for us. I want to see us talking deeply about these scientific questions. Excellent. With that, 2.5 minutes early. I'm going to hand it over to MacKie to give us a little intro about University of Florida and where we are. Oh, that's the wrong one.
Awesome, thank you. Ooh, I don't like that. Well, hi, I'm MacKie, for those of you who don't know me, and I'm an assistant professor here at UF, and I lead the Gator Glaciology Lab. So I'm so thrilled that y'all came out to Gainesville for this, and I wanted to give you a little bit of an introduction to where you are and mostly tell you about where to find gators, since that seems to be like a high priority objective for a lot of people this week. So this is our first southern WAIS, and I think it's, even though it feels a little random, it's important to remember that things changing at WAIS will have a direct impact on Florida's future. So the South has a lot at stake here, and I think there are some opportunities to make gains in the South and think of how we can better engage with this part of the country. We've got a pretty standard WAIS schedule with some talks, poster sessions, breakout discussions, and some dinners and excursions, which I'll talk about. I also want to highlight that on Wednesday morning we have an invited speaker, Dr. Megan Ennis from the Natural History Museum, and she specializes in climate change communication and outreach, so she's going to talk about her work and give some, impart some wisdom on us. There are also a few undergrad communications interns here who are attending WAIS and are going to do a little, I think, article or blog on WAIS science, so make sure to make them feel welcome. I've also been contacted by a number of UF undergrads who are cryo-curious and wanted to attend, so there are going to be some members outside of our community who are really interested in hearing what we're all about and hopefully helping disseminate our knowledge beyond just our community. Just to give you a little bit of orient yourself to where we're at. We're over here. This whole block is UF campus. So we're right across the street from the UF museums. This is where we were last night. Tomorrow night we're gonna have dinner downtown at the Woolly. And then these are some of the prime spots to see gators in Gainesville. And there are also some bat houses on campus, which are fun to check out. So this is Lake Alice. That's a big lake on campus that is full of gators. And this is Depot Park downtown. I think Depot Park is your most reliable gator viewing opportunity. I've always seen gators there. But yeah, so we're right across the street from campus. A lot of campus is pretty on campus. There's even a bear on campus over the summer. So if you're one of those people who likes to get up really early and go running, just be aware. This is Depot Park downtown, so there's a pond that has a few smaller gators that are always hanging out there. Riley and I saw these the other day. Yeah, if you just walk along the edge of the water, you'll find them. A number of people have been asking me about, like, what do you do if you see a gator? And really, like, not much. Just give them some space, about 20 feet of space. Don't stand near, like, the edge of the water at dark. So if you're at Lake Alice at night, don't stand on the edge of the water if you can't, like, see very well, because they can be swimming just beneath the surface. Don't touch them or do anything weird to the gators. They're generally most active at dawn and dusk, so that's when you're most likely to see them. And as a rule of thumb, you should just assume that every body of water has a gator in it as a default. So this is a picture of me and Dusty and a little gator in the background, demonstrating good gator safety. And, you know, as an extra precaution, you may choose to put a person between yourself and the gator as kind of an offering. So, welcome back, Dusty. Next to Lake Alice on campus, we have the UF bat houses, which is one of the largest bat colonies in North America. They eat billions of insects every night, so we like them. And like about 10-15 minutes after sunset, you can watch them all emerge, which is like, it's pretty cool to see what hundreds of thousands of bats look like. Make sure that you're, giving yourself some distance from the bats. Don't make any noises to interfere with their geophysics. If you see a bat, on the ground, don't touch it, or you'll need a rabies shot. It also might be a good idea to wear your WAIS hats because of falling droppings. But anyways, that could be a really good pre-dinner activity if you're interested to see the bats. We're right across the street from the Florida Natural History Museum, which is awesome. The Natural History Museum is free. So that could be a good Wednesday afternoon activity when you have that break. The Natural History Museum also has a butterfly garden that's not free. I think it's $14. But anyways, that's well worth a visit. The Natural History Museum is also next to the Harn Art Museum, which is also free and has a lot of like Florida art. If you have some extra time here and want to go hiking, I recommend checking out La Chua or Sweetwater, which are probably your best gator viewing opportunities. Devil's Millhopper Geological State Park has this big like sinkhole with a boardwalk going down into it. Also fun to see. And then we have Payne's Prairie that has like bison and horses and stuff, and gators. So yeah, please let me know if you're looking for things to do around town. But anyways, I'm so happy that you all made it out, and I hope you have a really fun and unusual WAIS. Thank you.
Hi, everybody. Our first session today will be piecing the puzzle together. I'm Ryan Venturelli.
I'm Paul Summers.
And we'll be convening this session. There will be 3 talks. We will have time for your burning questions as we're swapping between slides. So if you want to ask something, please just walk up to a microphone so that you're ready at the end of a talk as we're swapping those slides. But we do have a full 30 minutes for discussion after these 3 talks. So I'd like to encourage you all to ask interlinking questions during this discussion period. So as you're listening to these talks, think about ways that we can link these topics together so that we can maybe piece the puzzle together. Anything to add? All right. So on this journey this morning, we are going to transition from deep sea offshore drilling to subglacial West Antarctic drilling with our next talk from Brent Christner, whose home is right here at the University of Florida.
Good morning, everybody, and welcome to Gainesville and UF. My name is Brent, and I'm a microbiologist, and I'm really excited to have your attention on the first morning of this workshop to tell you about some results from two projects that I've just been fortunate enough to be involved with over the last few decades, as I think what I'm going to tell you has particular relevance to this audience. Let's see if I can try the digital. Try to take the plunge here. Yeah, okay, I'm going to really try this, guys. These projects both focused on a set of lakes that are at the confluence of the Mercer and Whillans ice streams, and roughly about 100 kilometers from the grounding zone. And these lakes are just a couple centuries old, and they are fed by a hydrological system that generates these fill-drain cycles. And when these lakes eventually drain, they discharge their water and microbes and biogeochemically transformed solutes and gases to the marine cavity below the Ross Ice Shelf. So a major objective of both of these projects is trying to understand the interactions between the lake and the marine system. And I'd say one of the key findings from these projects is that this has occurred in a time frame much more recently than we previously understood. The implications of this to the biology, well, I'll summarize about 15 years of research in saying that over the course of this period, we've identified about 12,000 species in the lakes. There are no gators in these lakes. We've currently found nothing beyond a single-cell organism, and that doesn't have any reason to think they're not cool systems. They're absolutely cool systems, which are driven, the organisms in them are deriving their energy from organics that are stored or processed in the sediment, as well as substrates that fuel lithotrophic metabolisms that are generated through water, rock, and mineral interactions. Now, the basal ice itself, as it melts, it's the water source for the lakes, it's the source of oxygen that's dissolved in the waters, but the microbes that we see in the overlaying ice are exceedingly rare in the lakes. We've also had an opportunity to study the marine portions at the downstream portion of the Wizard grounding zone, as well as other locations under the Ross Ice Shelf. The lake communities are completely different than these marine communities. I'll show you that we can even find, when we find groups that are common to both systems, just showing you examples down here of bacteria and archaea. Hopefully you can see this. The organisms we see from the lakes, which are shown with these blue branches, always groups differently from those that are from the marine system, which are shown here with these red branches. So different organisms. So these observations have led to the working hypothesis: if organisms that are in these lakes are not coming from the melting ice, and they're not necessarily introduced from the marine system, this is fueling our working hypothesis, the idea that the organisms that have colonized these lakes, they have originated upstream and have been transported into the lakes, co-transported with water and sediments, which are moving in this active subglacial hydrological system. And the implications from this are that these lakes are just one component of a larger scale series of ecosystems that are connected by dispersal or meta-communities. So because the ice sheet is a marine ice sheet, when the ice sheet disappears, That would mean that the evolutionary signals are in these communities would be erased, or maybe more precisely, are reset. So it's from this perspective that when a paper was published last year in Science, Lau et al., I got a feeling most in the audience are familiar with this, we read this with great interest. Those of you not familiar, this is a data set that was generated from a species of octopus which lives all around Antarctica based on hard-won samples collected over decades. In this analysis, these authors concluded that based on trends of allele mixing between these populations, that the best explanation for the time frames that they estimate is that for these octopus populations to mix, the ice sheet had to have collapsed. Now, with microbes, they reproduce asexually. So we can't use the same methods that Lau et al. Implemented in their study, but what we can do is we can leverage molecular clock theory. So this is a concept which recognizes that a nucleic acid sequence, if there is a mutation, this occurs at a random location. But the rate at which mutations occur is at a relatively constant rate. And what this means is that if one knows the difference between two sequences in a population, the divergence time from which their divergence times can actually be estimated. Well, in practice, about 60 years of data have shown that rates of molecular evolution vary widely between different organisms. They also range widely between parts of the genome that you look at. It also widely depends on environmental conditions. And if there's actually time in the discussion, I can expand on this and how this is actually really probably important in a subglacial environment. So any mutation rate that we might pluck from the literature and assess these data with is most certainly going to be the wrong value. But I'm here to tell you today, for the purposes of this discussion, that actually doesn't matter. First of all, what I want to do is to share with you some data that was collected, oh, sorry, that looks so awful, that was collected with our collaborators at COPRI using amazing technology. This is technology that allows us to take an individual cell, pluck that genome out of that cell, sequence it, and analyze it. And we've done this, we refer to these as single amplified genomes, or SAGs. So in SLM, we've been able to pluck about 1,400 of these genomes from the water and sediment, representing, from our analysis, looks about 200 new species and about 4 new genera. So that in itself doesn't necessarily mean that these communities are novel, but I do want to highlight some of the novelties by highlighting a group called the Nitrotoca. Now, unfortunately, in this phylogenetic tree, this is very difficult to see, but this is a group of bacteria that we know, based on their gene content, will oxidize organics, but it also has the capability to oxidize a cadre of lithotrophic substrates. If you're going to engineer something to live under an ice sheet, it might look like Nitrotoga. Well, what I would like to be showing you here, and this is very difficult to see, Nitrotoga is shown here. These gray branches and gray circles, which I'm sorry are impossible to see, represent the genomes that are related to this organism that are in a database that represents what we understand to be the current phylogenetic breadth of bacteria. These Nitrotoga taxa, they cluster exclusively by themselves. And I want to show you this trend on a larger scale within the data. Over here, these are average nucleotide identities. This is us comparing all the SAGs that we've analyzed to those in the genome. And I do understand those are very difficult to see, but what I would just want to show you down here, that these values that we see from the lake, they are much lower than we see from cells that have been recovered from the Ross Ice Shelf. So this implies not only at the amino acid nucleotide identity level, but also at the amino acid identity level. This is essentially our prediction of what these proteins look like. They look very different than things in the database, and they also appear to be more diverged than those in the marine system. And so this doesn't look like a group of organisms that might have just been introduced into the ice sheet a few thousand years ago. I want to provide you with just a little context of how we might look at this data and think of it in the passage of time. And it turns out that we know most about evolutionary time and divergence in the bacteria that make us sick. I want to show you a case of an organism called Clostridium difficile. Many of you may know it as C. diff. In the past 3 decades, it's emerged as one of the most important pathogens that we deal with, but it didn't come to be 3 decades ago. It has a path of evolution that stretches back to a point when all humans, bonobos, gorillas, all shared a common ancestor. And over a course of about 13 million years, it has diverged into groups that have geographic and host specificity, but it's never diverged upon a point of about 95% average nucleotide identity. To get at the kinds of levels of difference that we're seeing between our genomes and those in the database, we come to another group of organisms, some of which, even if you're not microbiologists, you might know the name of: E. coli, Salmonella. By the way, these are organisms that are in the same phyla as Nitrotoga, the group I just highlighted in subglacial Lake Willen. So not completely different bacteria, but we know that these organisms diverged about 140 million years ago. They share about an average nucleotide identity that we see in our genomes with respect to those in the database. So I want to leave you with a few key points here, but I do want to emphasize that all these assumptions are based on the fact that there are no obvious freshwater subglacial refugia for these communities if the ice sheet disappears. That's me saying it. Actually, if this is an audience that has that knows about possible refugee that these communities might hide in if the ice sheet collapses, I would be really interested in those discussions. And I want to also emphasize we really have no handle on the mutational rates of organisms that exist under ice sheets. But the levels of diversions that we're seeing in these communities, they would be of hundreds of thousands of years. These really look like more ancient isolations. And with that, I will acknowledge a bunch of people that are really involved. Many are in this audience in these deep drilling projects. Thank you.
All right, there we go. So is the implication that the ice sheet didn't retreat this far to these lakes?
Well, so if there was retreat that didn't collapse the ice sheet, it doesn't fully explain our data if the interpretation is that these things are coming from upstream. Mm-hmm. So that is, the ice sheet doesn't have to disappear for marine carbon to be introduced most recently, and that's what they've used. Those recent introductions don't seem to have, like, there were definitely marine organisms that would have been introduced by those timeframes, but they are no longer there in quantities that make it look like a marine system. Looks like a system that is coming downstream versus upstream. I'm not sure if I'm answering your question, but.
No, I think you are. So essentially, any marine organisms have flushed out of the system, and what's left is coming from upstream in a place that the marine ice sheet did not retreat to. Is that correct?
Yes, I would say that is a correct interpretation of how we're seeing it.
Yeah, very interesting.
Brent, how about in the down core pore waters? Is all of the lake material lost at the grounding line, or as you go further down into the cores, do you see any trace evidence of a previous lake, or vice versa?
Right, okay, so I can't really comment on that in the grounding line core, but what in the Mercer core, what I've really focused on in this talk was the portion of the core that we know is actually the lake biota, the stratified layers. The deeper portion of that, that is either marine origin or more representative of the sediments that are underneath the ice sheet. I will say that there are probably more marine taxa that we can identify in those deeper layers than in the lake layers, which does make sense if that's essentially been a bulldozed area.
So our final piece of the puzzle today will be relatively recent, just 8,000 years ago, by Roger Creel from Woods Hole Oceanographic Institute. And do you know how to make it go? I'm new to this. Do you know how to make slideshow? Someone knows. View. Thank you. There we go. Thanks very much.
All right. Thank you all very much for. So, the 8.2K event. Think of it as the most recent pre-industrial shock to the climate system. So, we're auto-advancing. Okay. So, mental model for what's happening on Earth. We have the remnants of the Laurentide ice sheet that are sitting on Hudson Bay, and there's a saddle of ice that's connecting domes of the ice sheet, blocking proglacial Lake Agassiz Ojibwe from flooding into the North Atlantic. Around 8,200 years ago, that flooding happens, sending fresh water into the North Atlantic. And when you flood the North Atlantic with fresh water, as best we know it, you slow down ocean circulation. So the question that I'm going to address is what might have happened to Antarctica when this freshwater flooding occurred. So I'm going to walk through a couple premises for a mental model for what might have happened. So premise number one, when the 8.2K event slowed ocean circulation, the Southern Ocean warmed. And this premise is based off of quite a bit of what they call hosing experiments, dump fresh water into the North Atlantic in an ocean model and see what happens. What typically happens is that the Southern Ocean warms by about a degree. Here are three other studies showing, with different color maps, but showing a similar thing. Premise number two. As the Southern Ocean warmed, circumpolar deep water penetrated the Antarctic shelf. This is happening now, and it has to do with a bunch of different factors in the Earth system, including potentially westerly speeding up, moving southward. But what happens is you get warm water that otherwise would be off the continental shelf, intruding onto the continental shelf and eating away at the base of the ice sheet. Premise number three. When circumpolar deep water penetrated the Antarctic shelf, the Antarctic ice sheet thinned. So there's quite a literature on Antarctica thinning in the Holocene because of ocean influence. So West Antarctic ice sheet retreat driven by Holocene warm water inclusions. Ocean is the main driver of Antarctic ice sheet retreat in the Holocene. We could just keep going around in circles looking at these papers. So the motivating question for this talk is, how did the 8.2K affect the Antarctic ice sheet? And we're going to look at this from three independent sources of evidence. One, an ensemble of ice sheet simulations from the parallel ice sheet model. These are from Albrecht et al. 2020, 2021. Second, cosmogenic exposure age thinning profiles. And third, marine sedimentary data. And all these are going to feed into this picture of early Holocene Antarctic change. Okay, so first, the ice sheet ensemble. We have a paper coming out pretty soon looking at Holocene global mean sea level. I presented on this last year. And as part of that, we reconstructed rates of Antarctic thinning over the Holocene. And one thing popped out, this is in like supplemental figure 37 or something, that there's this spike in thinning rates right around 8.2. So this was a motivation to say, what's going on there? So we changed the weights a little bit to look at this more in detail and came up with a modeled rate of Antarctic contribution to global mean sea level. So this is ice volume above flotation, a little bit more nuanced than that, but basically above flotation. And the forcings that are going into this parallel ice sheet model, we have sea level forcing, only global mean sea level. They're not using glacial isostatic adjustment. Air temperature forcing from waste dome C, and ocean temperature forcing, which in this modeling setup was a smoothed and lagged version of air temperature. So crucially, this model is not seeing proxy ocean temperature, it's only seeing versions of air temperature. And what popped out was that the sequence of Laurentide ice sheet collapse events coincide with peaks in Antarctic thinning. So here's ice saddle collapses of the Cordilleran ice sheet, Inuitian ice sheet, Laurentide ice sheet, really nicely. Each of those color bars are from essentially radiocarbon dated reconstructions of Northern Hemisphere Laurentide behavior. In this model, the Northern Hemisphere is talking to the Southern Hemisphere, and it's probably talking through air temperature because that's really the only, aside from global mean sea level, that's the only forcing that the model is seeing. But the 8.2K event doesn't coincide with one of these spikes. It's a little bit after one of the saddle collapses of the Laurentide ice sheet. So if we're only thinking about air temperature, 8.2K event is not coinciding with Antarctic, at least modeled behavior. So let's then look at the cosmogenic exposure age record. Okay, so for folks in the audience who don't think about Cosmo all the time, I'm with you. Until 4 months ago, I didn't either. But to give you a primer for Cosmo, energetic particles are bombarding the atmosphere at all times. If they hit quartz-bearing rock, they cause chemical reactions that produce, among other things, beryllium, aluminum. And so if an ice sheet is covering rock with enough ice, that rock is shielded from this process. So as the ice sheet thins, the rocks at the top of the mountains start accumulating beryllium faster than the rocks at the bottom of the mountain. So you can then, by doing chemistry on these rocks, sampling them, tell a story about the exposure history and therefore the thinning history of a mountain sticking out of the ice. Okay, so I have done none of this field work, but other people have, and it's really good. So we're gonna zoom in first to West Antarctica, just to give you a sense for what these data look like. West Antarctica, we're gonna zoom into the Pirit Hills, and now we're gonna stand on the ice and look at the mountains. And then looking at the mountains, we're gonna climb up one of the mountains, and there are boulders on this mountain that folks have sampled. Folks then took those samples back to the lab and produced these beautiful data in elevation above present-day ice. So our intervention on these data was to fit them with penalized cubic splines, similar to, if you're familiar with Richard Jones' ICE-T tool, but I had trouble even getting it to run, so I rewrote it in Python from scratch and modified it. Not all the data are quite as pretty, so we had to do a bit of work on them. But there's a database of these data called the ICE-D database. So there's actually quite a bit of data from all around Antarctica. So we took all these data and produced continuous thinning rates of Antarctic change over the Holocene and stacked them. And when you do that, so now we're looking at percent of sites thinning and thinning rate on the with color bar. When you do that, one of the things that pops out is that there's rapid thinning that coincides with the 8.2K event. Mapping the Laurentide collapse sequence on this, there's also correspondence with the Laurentide. It's not quite as pretty as the model if you split this out into sectors, one of the things that's interesting is that there's a significant amount of thinning in particularly marine-based sectors of the ice sheet after basically any substantial ice melt in the northern hemisphere is happening. So Greenland, we could discuss about exactly what's happening 7,000 to 6,000 years ago, but it's not major collapses. Nevertheless, in the Weddell Sea, in Amundsen Sea, in Ross Sea, there's continued fast thinning in the mid to late Holocene, and there's also thinning right around 8.2 Ka. And this aligns with what we know from marine sedimentary data. Think to interpret this plot, think your standard oceanography plot where you have wiggles stacked on top of each other. But if we stacked 30 of those plots, this is what that would look like. So I took marine-based proxies for ocean change and recalibrated them to present, like InCal 20, Southern Hemisphere 20, radiocarbon timescales, and then where the original authors had identified change happening, so ice discharge, open sea ice, high current speed, high productivity, circumpolar deep water intrusion. That's where these uniform probabilities start and end, and then there's some uncertainty on either end, but this was just a way to say, kind of in aggregate, what do we know about ocean variability and how it's influencing Antarctica in the Holocene? And the picture is we start around 9 and it keeps going until 6 or even a little bit after. Which brings me to the takeaways from this talk, which is that on the modeling side, we have an ice sheet that's being forced basically only by air temperature, and we get really interesting correspondence with northern hemisphere abrupt ice change events, which suggests there may be an atmospheric teleconnection driving the thinning in at least the ocean, at least the ice sheet model, and given that this also shows up in the thinning rates, probably actually in reality also. That atmospheric forcing is not producing the 8.2 Ka thinning that we see and the mid to late Holocene thinning, which suggests that we need to also be thinking about ocean forcing as a dominant driver. This is not a new hypothesis in the field, but it's worth ice models to be reproducing how Antarctica actually deglaciated. I think we need to be really pushing using marine proxies for ocean change rather than scaling air temperature, because circumpolar deep water is not intruding onto the continental shelf, is not necessarily showing up in WAIS Dome C, but it's certainly having an effect. So with that, I want to thank my collaborators on this project. Science is a team sport, so none of this happens just because of the person standing at the front of the room. It's always a team effort, and thank you very much.
I'm a little smaller than Roger, so I need to adjust this. If we could ask all of our speakers from this session to come up and join us in the front of the room. Thank you, Roger. That was thrilling. I think we should just leave this up because I like the gator. So just to walk us back through this puzzle we're piecing together, we started offshore with iceberg-rafted debris, and then we moved onshore thinking about microbes underneath the ice sheet. And then we finished up thinking about how rocks in place above the ice sheet can tell us about things that happened about 8,000 years ago. Would anyone like to start us off?
Thanks, Brent. That was a really interesting talk. I'm curious, could you comment on the decontamination protocol, especially if you're going in between lakes? That's obviously a significant challenge with any kind of microbial work.
Yeah, We spent about 3 years figuring out how we were going to do this. The drill that we're using, the water, we're able to keep it very clean by circulating it through 100 gallon per minute level filters. That's down to 0.2. We also heat the water up, so anything that squeaks through those filters and actually goes to 95 degrees Celsius. Most Antarctic organisms don't do well. So essentially we're drilling with pharmaceutical grade water, and the data that I haven't shown you is not only when we drill into the lake or we're collecting water in the sediment, but we're at every point sampling the water, cleaning all the instruments with 3% peroxide. I can point you towards a couple, not really riveting papers on how we did this, but it's pretty well documented.
Okay, thanks for really 3 terrific talks to get us started. And I just wanted to make a comment and some discussion about how the WAIS community really benefits from understanding processes, modern processes, and the perspective from the paleo record is what really brings it all together. And that's one of the great things about this community, seeing proxies for how the ice had changed over millions of years is really important for understanding how the ice sheet has changed over much shorter time scales and might change into the future. So just as a point of discussion.
So Mike, you kind of blasted through the models and showed some pictures, and the thing that struck me was the Wilkes Subglacial Basin deglaciated along with West Antarctica. And then we can look at kind of synchronization between basins with Roger's work, and the microbes tell us a little bit about what might have completely collapsed, what didn't. And so I'm wondering if we can actually start to distinguish which marine basins are actually collapsing with more granularity, rather than just there was a bunch of sea level that came out. Like, what is stopping us from this now?
That goes pretty far. I'm not sure I can answer that. I mean, the model we used for this ice sheet model in the Ross Sea and the Weddell Sea, that was it's a modified Penn State model that is coupled to a CESM Global Earth System model. I didn't do it, a postdoc of Axel Timmermann did it, and we were quite surprised. We knew earlier on that right around 2 million years, the ice sheet started to build up more extensively in the Ross Sea, but that was just a little bit, if you see it, a precursor, and then it dipped down again, at least in the model, to like 1.5. And then from 1.5, the ice volume really went up like a lot, you know, and then it took like maybe 500,000 years to come to that first culmination point. And then in the Weddell Sea, apparently all of that happened in a much shorter period. What we had before the question we asked is we didn't see any strong response to orbital forcing in the IBRD signal. Although if we look at papers from Tim Nash, you know, they said that we had the 40,000 year cycle. We look at other papers, they had eccentricity precession forcing. We do see bits and pieces of that. Quite a few of these papers only looked at short meaning just a few 100,000 years, maybe in the Pliocene. But if we look at the whole long picture, there isn't really a lot of consistent orbital forcing until we reach about 900,000 years. So we were wondering, what is that? And this ice sheet model showed that right around there, that's when we have a large buildup of the ice sheet, and that's when it goes into these large glacial, interglacial fluctuations. So at least we can say, okay, only when the ice sheet is at that global rhythm, you know, there's a big drum playing, that's when we see the response to orbital forcing, which leads us to the conclusion that all the response before 900,000 years, or at least before 1.5 million years, is internal ice sheet dynamics, not so much connected to any global forcing. And that is, in the model, differentiated between at least these two basins. We would need to discuss this with Axel Timmermann for details.
Well, another aspect for Matt's question is provenance of the IRD.
Yeah.
And we can in many cases, tie it to sources.
Yeah,
And
and
so,
that's-
for example, in the Iceberg Alley and 382 and then 396 in the Amundsen Sea where we drilled, there was one very distinctive kind of green sandstone, which could be tied to the Whitmore Mountains, that was found out in Iceberg Alley, but also in the Amundsen Sea. And that's really compelling evidence of a specific source coming, indicating
Yes.
those basins.
I mean, that's what we do right now. We have taken all the samples to do these provenance studies, and I just hinted on that. This northern side probably has more influence coming from the Ross Sea, because that's when the bergs go astray and are directly incorporated in this system. And the southern side is more from the East Antarctic ice sheet. And we do have indication. We had a pilot study out, a paper that already showed that, and all the samples have been taken, but we face a big problem. We don't get it funded. I tried 3 times to get that provenance work funded. I failed. Sidney Hemming is trying. We have another postdoc which deviated to Austria now. So all the people in the community know how crucial and critical that data is. The reviewers don't. They say, oh, that's not important. We don't know anything. No one, and they don't fund it. It's really a big problem. And that is tedious, very expensive work. You know, it's not just, this data set takes a lot of manpower, a lot of argon dating multiple times. That's all expensive. You need to have several tens of thousands of dollars to make that happen. Nobody gives us that kind of money. That's just a problem.
Hey, Mike, I hear some action here. We can get that in the waste science plan.
Yeah?
Put it in the Google form.
Yeah.
Microphone, please.
I should know better. Right, you're connecting a change in state to volume. It expands, we get this global synchronization. It contracts, does it go back to where we just see this internal variability again? Like, where's that threshold? That's important for us, right?
Yeah, I mean, we don't know yet, but it's there. Yeah, it's threshold. It works all. That's what we learn. It all works through thresholds. Yeah.
Thanks for your talks. So Roger, near the end of his talk, mentioned this paleo to modern connection, how using the data from the past to inform what we need to do to know more about the future. So I'm curious if there are data that you're aware of that have already been collected that you think revisiting would be helpful, kind of like how Roger used ICE-D to kind of reanalyze some of these questions and think about where we could go. If there are data that we could reanalyze to get at modern questions, or what data would be really useful to gather now that could inform future behavior.
So the microphone's over on this side, so I'm gonna answer that. I mean, there's a lot of support right now for doing this sort of legacy data projects that, for instance, Ryan has funded to do, to take existing marine sediment cores and date them better and do more analyses on them. It does, speaking as a modeler, it does seem like anytime we can add high-resolution analyses to cores that already have information about them, it expands our knowledge more than, for instance, taking a core about which we know nothing and doing low resolution, because I think the multi-million-year perspective still requires high-resolution sampling. As you were talking about this threshold, I was thinking, well, given connections between the northern and the southern hemispheres, it might be that actually the answer to why does Antarctica start going up and down is also a northern hemisphere question. And so therefore, paradoxically, to understand things about Antarctica, we actually need to go to sediment cores, for instance, around the Laurentide ice sheet and figure out when does the Laurentide hit a threshold of waxing and waning, and because there's another marine-based feedback loop. So I think it's a global problem.
Just to add on that interhemispheric component, we work on that for like more than a decade. We had a paper out in 2011 in Science about that, where we proposed that an initial deglaciation, I'm talking now just the last deglaciation, right around 19,000 years, triggered a similar deglaciation in Antarctica. But back then it was just a snapshot of a model, of a gravitational model into that time sphere. And now we have, in 2020, we published another model with Natalia Gomez, and that was in Nature, and that showed us clearly that through the deglaciation, what happens is the northern hemisphere with this big ice mass volume loss, that triggers the millennial scale ice mass loss, mainly in Antarctica, specifically during the 3 meltwater pulses, 19K, 1A, and 1B. So the fate of the Antarctic ice sheet mainly relies on what the northern hemisphere ice sheet does, because if you lose a lot of ice mass over there, you trigger a sea level increase near Antarctica, and you trigger it spatially variable. And that spatial variation is more or less what we see if we collect the glacial data. That pauses through, so there's a very logical model. And looking into the future, Natalia Gomez had just a paper out this year in Science Advances, which really takes that to the next level and says, if we are to lose ice in Antarctica, what happens is we have a rapid uplift. So the amount of water we put in the ocean determines the relation between that uplift and the sea level rise. So if we go to lower emission scenarios in the future, that uplift might outpace the sea level rise and contribute to AIS stability. That's a very important message. A few years ago, we were much more pessimistic, saying like, once that ice sheet is in a runaway situation, it will never recover, at least not on socially relevant timescales over the next decades to centuries. But this has changed that view, mainly because one thing happens. There are no big northern hemisphere ice sheets anymore. There's Greenland, but that's a little player. But the Laurentide ice sheet is gone. So there is hope that in peak warm times, and we do this now for glacial termination studies back to the 600,000 years. We study the last 6 glacial terminations. And we always see this apparent stability of the ice sheet during warm times. It's early on when sea level is up. That's when it's stable. Of course, with the CO2 situation right now, we go out of that system. How far, we don't know. That's what we need to talk about. But there is hope at the horizon that there are, again, thresholds, stability thresholds in the ice sheet that might keep it stable or might not lead to a catastrophic loss. Sorry, I talked too long. You already told me that before that I didn't stop.
That was great, and I think that that is actually a really great place to push Brent to get involved here. I think Brent presented some really compelling evidence that microbes can be pretty good indicators for lack of West Antarctic ice sheet collapse. So in the case where we need an indicator of these thresholds or these extreme scenarios that we can't get from other indicators that Roger and Mike have already named, Brent, do you have any ideas how the bugs can help us out here?
Yeah, well, I mean, one of the ways they've helped us out, I think, with the problem is that because they look like they're coming with water and from underneath the ice sheet, it means that I think if we want to understand the relationships, not just between the lakes we've studied, but the larger region underneath the ice sheet, I think we're actually talking about going to those places and drilling in and sampling them. Okay, in the absence of that kind of new activity, the kinds of things that we could still learn, I pondered on the idea that I put a bunch of question marks at the rates at which these bacteria in these systems mutate. It is actually possible, without getting new samples, to take bacteria that we have recovered from these environments and do very similar kinds of experiments. So it doesn't necessarily require a $30 million drilling project to get at this question. And I'm giving the case of microbes. I think also that my intention really wasn't to dump on the Lau et al. Story. I think it's actually a fascinating story. It's hard for me to fit our observations in with that. So in a similar way that octopus populations have informed this, I think there are other aspects of the marine community that can inform it as well. If, for example, if these patterns are found in octopuses, which have limited distributions, they should also be found in other organisms that have very similar patterns.
So I have a question also on the microbes. So can you give any discussion on any metabolic function of the microbes that are peculiar or that can be used in other aspects of science that you've discovered? You mentioned that they use a soil, and I know the soil has like high content in specific metals or minerals.
Okay. The one feature of, maybe not necessarily the microbes, but maybe one of the unique sources of energy that has emerged in studies of the subglacial system is that we know that if there's anything stored in the sediments and the microbes can get at it, they will. The rocks, which can have minerals in them, which microbes can access if they're in the middle of a rock, the physical action of the ice sheet actually helps that, pulverizing that material so that microbes and water can interact with it. And there's actually a third mechanism which we now understand might provide a wide range of not only mineral substrates, but also organic substrates. And this has to do with rock crushing. Some of you might be aware of this. This is sort of, in a sense, it's sort of old wine in new bottles, in that it was initially observed after earthquakes, that in these fault zones, they'd be chock full of high amounts of hydrogen. Like, where's the hydrogen coming from? Turns out you create these, oh boy, here, okay, my very small understanding is that you shear these silica minerals and you create free radicals. When they mix with water, it reacts with anything. So you can create ammonium, you can create methane, you can create acetate by crushing rock and adding water. So that microbes, I guess my message might be that in the system, microbes are taking advantage of every available resource. And the reason why they're probably microbial systems is that anything other than a single cell couldn't actually function on that flux of energy.
So I guess my question, another question, how can we take advantage of the microbes, right? Because I guess, some ideas is like, EV batteries are really popular now. It's like, we're progressing to a society where we need these high, valuable metals and stuff. So you think could be a link that we could discover in genomic function of the microbes like that?
Yeah, I'll try to give you a very specific example. These microbes that live under the constant threat of freezing have evolved proteins that interact with the ice in certain ways. It can actually help initiate the formation of the crystal. Once the crystal forms, it can actually bind to the crystals in a way that control the crystal in the way it recrystallizes. And so we're involved in projects now that are actually bioprospecting these compounds with the idea that if you have, for example, in the Arctic, you have a surface which is unstable because of warming temperatures. Are there compounds that can be added to strengthen that ice? Are there compounds, if you have frostbite, can you smear something on your face and it have one of these microbial compounds? Most of you who work in the Antarctic know that these are often referred to as antifreeze proteins. And these were first described in the nototheniod fish that live in -2 seawater, summer and winter. The problem with those proteins in wide-scale use is that they're from fish. So fish allergies are actually a big concern. Putting fish products in foods that humans consume or put on, that is actually a problem. And they're also the kinds of proteins that are really tough to make in large quantities. Basically, you need a room full of fish about this size to get enough proteins. And growing a room full of bacteria this size is actually really easy.
I'm still thinking about the bugs. So you said there has to be a refuge for these freshwater bacteria somewhere. and that has to go back hundreds of thousands, million years. So we can't have full collapse. I guess some of this is like, what does collapse mean? And that's a different question. Different people have different answers to that. But I'm thinking more pragmatically, if we have these $30 million drill projects, and we can start taking transects and looking at the genomics along transects up to the divide, what do we get out of that?
What we would get out of a transect to the divide? Well, okay. I guess one thing we could do, if we're upstream of what would be the catchment for the WAIS and Miss Lakes, we could essentially test all the hypotheses that I've just described. It seems like if we're moving towards the divide, we're getting out of that catchment. So now the question is, I mean, I made a statement based on 2 lakes that we sampled near the coast that like this could essentially mean there's a subglacial biome underneath WAIS. There very well may be, but they could be separate universes that don't even know either exists. But if they're connected, I think that tells us something else about the history of the ice sheet. As far as the refugia, I would just love I'd love to know more about this. I see the images like I showed in my talk, some artist rendition of what it looks like. I don't know. In the case of SLM, we do understand that there is some amount of source water from East Antarctica. I don't know if East Antarctica could be a refugia for the kinds of systems that we're talking about. Willans, no. Mercer, yes. So it doesn't quite fit the bill.
Go ahead. So this is following on with the bugs, and it might be completely off base, but if we're doing the cosmogenic dating and we're gradually seeing the ice retreat, in the rocks, there are a lot of weathering salts and often mud on the bottom of them. Can these be places for the microbes to live that you can track with the cosmogenic dating, kind of going up the profile and seeing maybe there's bugs there?
I won't say no. I guess if we're talking about profiles that are in rock it's probably a little hard to say. So you're figuring out provenance of compounds in the rock. It might be really difficult if you're even finding microbes associated with them to say that they have the same provenance. Biomarkers, maybe, of microbes that were preserved at the same state. Again, what would that tell you? You wouldn't get genetic information, which is, of course, the kind of data that I'm thinking of. I think if you want access to those genomes, the best available places might be sediments rather than rock. So if you can go places where you can go back far enough to get old enough sediments, we know from marine systems, I mean, it's pretty routine. People are looking at finding microbes in sediments that are 20, 30 million years old. You add food, they wake up. It's like these microbes are in a retirement home, and they're just able to wait for millions of years. So the rock system you mentioned, I feel that might be difficult, but if there's on average 1 kilometer of sediments beneath the ice sheet, the age at which is whatever, I think that's probably the place you do that.
I'm hearing a lot of plugs for deep drilling in the WAIS science plan, so I would just like to encourage everyone to get your thoughts in to the Google Sheet. We have 3 minutes left of this question session, and I would like to bring it to the future. I think a lot of times in the paleo world, we motivate our work by saying we need to learn about the past to inform the future. But I would like to know from all of our speakers today who talked about the last 1.5 million years, the last 125,000 years, and the last 8,000 years, how your work specifically can help prepare us for the future. So any final thoughts?
Well, one thing that is interesting on societal timescales is we had a paper in 2021 in Nature.com, and we looked at the last deglaciation and how fast an ice sheet collapse can happen. We could do this in Iceberg Alley because the sites there, these precursor sites, they have a very high resolution, so our sampling rate was between 5 and 15 years. And we saw that 6 of these events happen within 1 decade. Before, people thought like this takes, you know, decades, centuries, and we could nail it down in principle to 1 decade, which is from one sample to the next, which says if we find a core that has even a higher resolution, if you remember how it was with the ice sheet in the Northern Hemisphere, eventually they found something that initiated meltwater pulse 1A within 1 season. So we could even go further down. But that's kind of a frightening scenario that you see that this is a sharp threshold. It doesn't take decades to develop. And it's equally fast when it stabilizes again. So it really is a big threshold, and that's important for the models in the future. Just one example.
Both scary and encouraging.
Yeah, I'll take a slightly different approach to that question in that instead of thinking what science I could produce in the future to help this problem, I feel like I kind of just gave an overview of what that might be. And I guess I might think more practically about the kinds of, you know, if you're thinking of a project that is, for example, a deep drilling project, and that's something that you want to happen in 5 years, like, you need to be organized now. And I guess my sense from the way that these big projects work the best is when you have the most science incorporated, right? So if I thought, like, by myself going to drill into a lake just to study microbes might be great, it is not going to be the kind of project that NSF is going to invest in to just do one aspect of science. So like I think the kinds of challenges when I look to the future, when I think about science at this level, I'm thinking about how I can incorporate my work with the widest range of science. I made obvious connections in my talk between how the microbes are connected to the hydrology of the system. And now I've got like a question that's bouncing around in my head about microbes in rocks. That are maybe not under the ice sheet, but exposed. I've dismissed it, but I'm actually, it's still, it might keep me up tonight. Anyways, I think these are examples of ways in which we can prepare in that, prepare to set our science in framework that it is actually relevant to other sciences that can be incorporated in these efforts.
Thanks, Brent.
So we have 2 ice sheets left, and we want to need to know how they're going to affect each other. And I think what's exciting about the past is that you have many examples when ice sheets affected each other, and we can start to get at how much melt you need to have from the Northern Hemisphere in order for Antarctica to flinch, in order for there to be response. So I think like 30 years of data collection feeding into a cosmogenic nuclide exposure age database is starting to be enough that we can see not just the big scale meltwater pulse 1A, 1B, 19Ka, but also in the Holocene, as Laurentide is doing, still collapsing but on a smaller scale, that Antarctica is responding. So I think pushing forward, the question is like, basically, how much does what we have left is Greenland? How fast does Greenland have to melt in order for that melt to rebound to Antarctica? And can we go to times in the past when both Greenland and Antarctica were melting fast, and how do they affect each other? Because it's not just one or the other. So I think looking into the future, I'm interested in we could call it a threshold of interhemispheric connection.
Awesome. Let's thank our session 1 speakers again. And next up, Allie Banwell will get us rolling with our poster introductions.
Thank you. So now it's time for the 1-minute poster pitches. I'm Ali Banwell. I'm currently a research scientist at CU Boulder. Matt asked me to do this on the basis I might be good at dealing with some potential chaos, so we'll see how that goes. If I could ask all the speakers to line up in the order that they are on the schedule, which is also alphabetical order, that would be great. And then after that, anyone who is new to WAIS, who wants to introduce themselves, who doesn't have a talk or poster, you'd be very welcome to also come up. So there's about 20 posters in total, I believe.
Did you put it in the slide deck? It's updated at 9:15.
Yes.
Hopefully the internet's fast.
It did seem to align on that.
Try that. There you go. Look at that.
Yay, nice. Okay, Imogen first, please.
Okay, hi everyone. I guess I get the privilege of going first because of my last name. So my name is Imogen Brown, and I'm a postdoc at Binghamton University in New York. And what I'm presenting here at this conference is some work that I've been doing at more of an ice distal marine site. I'm going to use this one. And so I'm interested in understanding how Antarctic bottom water formation changed during past warm periods. And this is important because today we're observing a slowdown of Antarctic bottom water formation. So my work is to understand how this may have played out in past warm periods to sort of understand the long-term trajectory and climate implications of this. And so I'm working at Site 1123, which is off the east coast of New Zealand. It's a sedimentary drift deposit. It's in the pathway of deep water boundary currents, which might be related to Antarctic bottom water formation. I'm using sedimentary XRF data, so X-ray fluorescence scanning data, to understand the changes in sedimentary processes. And so at this site, the sediment changes are related to the amount of terrigenous sediment input and also biogenic sediment, calcium carbonate. And so I'm just trying to work out what proxies I can use to reconstruct grain size and calcium carbonate. And over the long-term record, which is through the Miocene, I'm seeing an increase in grain size, which potentially indicates an increase in current related to more Antarctic bottom water formation as the ice sheets were developing. Cool, thanks.
Right. Next up, Annie.
Hello, my name's Annie Chang. I'm a second-year grad student with Dusty at Stanford. This is my first time at WAIS, so I'm very excited to meet everyone. My poster is about basically the effect of melt in ice and what that says about radar signals. So we've used ice-penetrating radar widely in the field of glaciology to study the ice. And the reason why we use radar is because ice is supposed to be invisible to radar wavelengths. However, if some people have been looking at ice, and melt does exist in like temperate ice, subtemperate ice, ice cores, all kinds of ice. There are little veins of water that still exists below freezing temperatures. And my work, my poster, is about, okay, if we take into account these little pockets of melt, they might be scattering your radar waves, or they might be attenuating or reflecting them in some way. So just looking at how that would affect your radar signatures. Thanks.
Hi, I'm Sam Chester. I'm a third-year PhD student at Columbia. and I'm going to be talking about a new model that I've been building, coupling the parallel ice sheet model PISM to a GIA sea level code written by my advisor, Jackie Osterman, and thinking about how we can use that model to test some different hypotheses for grounding line re-advance during the Holocene. So yeah, thanks.
Hi, I'm Miley Corso. I'm a senior undergraduate at Colorado School of Mines, and my project focuses on updating Antarctica's snow accumulation estimates by Global Navigation Satellite System Interferometric Reflectometry, which is a system of GPSs set up at a height above the Antarctic surface, and using satellites that orbit around that area, we can use signal-to-noise ratio by the direct and reflected signals to calculate the height at which those GPSs sit on, and by that we can use that to get some snow accumulation estimates. So I looked at the data taken from 2 different frequencies, and I was looking at the point-to-point variability to see which ones were providing the most realistic results. So, yeah.
Good morning, everyone. Hope I got that right. My name's Lucas. I'm a junior undergrad student working with Ben at Colorado School of Mines. And I've been looking at this polar gap airborne radar survey, which is flown in polarimetric mode. So we're looking at different polarizations of radar waves and actually using ice anisotropy and a little bit of cloud computing to compare those different polarizations and see what we can learn about ice dynamics and ice flow around central Antarctica. So I'm looking forward to meeting everyone. This is my first WAIS and my first conference. So yeah, definitely keep my poster on your radar.
Great. Thank you. Next up, Chris.
Hi everyone, I'm Chris. I'm a second-year PhD student at the University of Minnesota, and I'm advised by Lars Hansen and co-advised by Meghana Ranganathan. And I'm presenting on a phase field model that I've been working on, and it's with 2 different rheological models for the ice. One is linearly elastic, and another is a Maxwell model, so it's a viscoelastic model for the ice. And I'm looking at how that affects the rate of crack propagation in ice shelves. And so I'm also using 2 different geometries. One's with longitudinal stretching, and the other is with bending, like from tidal flexure. So yeah, I'm excited for you to see the results. Thank you.
Great, thank you. Sorry, next up, Teresa.
Hi everybody, this is my first WAIS, I'm excited to be here. My name is Teresa King. I'm working as a postdoc right now at the University of South Florida, so just a couple hours south of here. And my poster is going to be talking about how we can get the most accurate radiocarbon-based chronologies to get at these things like timings of events that are happening that we learned about earlier this morning. And so we use a very specialized advanced radiocarbon dating method called ramp pyrolysis oxidation, or ramp pyrox. You may have heard of it before. But because Antarctic marine sediments are very carbonate poor and tend to be also very low in TOC in a lot of the places, this method is really good at isolating that more labile, I don't really like to use that word, a more a younger portion of sediments that is included in that mixture of bulk organic material, so we can get an age that's closest to the true depositional timing. And what I've been working on is updating some of the protocols involved in this method and looking at our blank correction, which is something that's an inherent contamination that happens just from processing samples. So I wanted to create a space to talk about blanks, and if you are interested in this method, you can come talk to me to learn more. We can talk about blank corrections if you want to use it. I want to talk about things that you should be asking our lab and other labs that are coming online with their own dirt burner systems. So I'm happy to chat.
Great, thank you. George, next.
Hi, everyone. My name is George Liu. I'm a PhD student at Columbia University. Some things are a bit cut off, but yeah, so what I do is I have a background in ice penetrating radar and some basic ice sheet modeling, but there's been a lot of really cool recent work using these things called physics-informed neural networks. Some people here and people not here have applied them in glaciology in many cool ways, and I thought I'd give it a try. So my poster details my work thus far developing this new physics-informed neural network setup that I plan on using to infer for ice rheology at ice divides, using some previously unpublished PRES data from my advisor, Jonathan Kingslake. So yeah, come by and hear about my progress.
Hi everyone, my name is Michael Field. I'm a third-year PhD student here at the University of Florida, where I work with MacKie. MacKie and I have been teaching a class together where we're having undergraduate students invert for bathymetry beneath ice shelves using gravity data. So this poster has results from some figures that they've generated throughout this process at various ice shelves around Antarctica. And I will be speaking about this more in depth in my talk. And we might have some of our undergraduate students there, so please come check it out.
Thank you. Daniel, next, please.
Hi, I'm Danny May. I'm a PhD student at Stanford University, and I'm focusing on the eastern shear margin of Thwaites Glacier, which is shown at the top of this map of ice surface speed. And unlike the other shear margins of Thwaites, the eastern shear margin is not, or its location is not defined by bed topography, and so is sensitive to migration in the future. And so I am using seismic data that were collected this past year through ITGC, as well as existing airborne radar data to determine what subglacial or anglacial processes control where this margin is located. And specifically, I'm looking at looking at how seismic reflectivity and radar reflectivity change as a function of bed incidence angle, because this relationship tells us something about the bed composition. And so the goal of what I'm doing is to kind of map out how bed composition changes across the shear margin. So yeah, feel free to stop by, and thank you.
Hello, my name is Rachel Miney, and I'm fixing to start my PhD at the Colorado School of Mines with Ryan Venturelli, but in this poster session, I'll be presenting stuff that I just completed for my master's. So I'm presenting data on 4 sediment cores, all collected from Ross Bank. I am a geologist, but I like to use geologic tools to study the behavior of ice in the past. Specifically, the proxy that I use are diatoms, and so come to my poster to learn more about how diatoms can be used as paleo records, to learn how about those can give us inferences about how ice sheets, specifically on pinning points, behaved during the last glacial maximum.
Great. Ellie Miller is next.
Hi everyone, my name is Ellie Miller. I'm a second-year PhD student at Colorado School of Mines working with Ryan Venturelli, and this is my first WAIS, and the title of my poster is Quantifying Subglacial Groundwater Exfiltration in West Antarctica Since the Last Deglaciation. And my collaborators are included on the slide as well, all of which are sitting in this room, which is really exciting. And this project was born from a recent Roble and colleagues paper in which they quantified groundwater exfiltration of the Antarctic ice sheet over contemporary timescales. And as a paleoglaciologist, I've been thinking about longer geologic timescales, so I've spent some time combining a few paleo data sets, many of the same ones which Roger mentioned, to sort of get at what groundwater was doing in Antarctica over the past 20,000 years. So if you want to check out some preliminary results from this work, come see my poster. Thank you.
Thank you. Joe is next.
Hi everybody, my name is Joe Ruggiero. I'm a PhD student at School of Mines, and I'm Ryan's third student who's giving a poster here.
This is my first WAIS, and my PhD research largely is focused around thinking about how radiocarbon is transported around the Southern Ocean. And when we date sediments with bulk acid insoluble organic carbon, this manifests as a large offset often in ocean sediments. And so my first project is focused around looking at core tops and thinking about why some core tops are so old. In some places, such as the Ross Sea, we have ages up to 20 to 30,000 years too old. And so I'm thinking about some sedimentological processes and geological and glacial processes that contribute to this erroneous age offset. So if this is interesting, I'd love to chat and hear your thoughts. Thank you.
Thank you, Matt.
I will have a poster. I get to deal with bad formatting issues now. I'm presenting a poster on behalf of the ISAT-2 Science Team Land Ice Working Group, which I co-lead with Michaela King. And so this is basically just an advertisement for what can ISAT-2 do for you. We collect photons. You can see here. You see lots of really cool papers coming out using data down to the photon level, but that's not all we do. We spend a lot of time developing this whole set of really easy to use data products from ISAT-2, so anyone in this room can use ISAT-2 data. So it goes from individual photon level data all the way up through gridded maps of ice sheet change. And so these are figures, the code for these figures, there's actually a QR code you can scan and get to the Jupyter Notebook to generate height change figures for both ice sheets in 10 minutes or less, right? Like, these are incredibly easy to use data products that everyone should be using. This is the most up-to-date information about ice sheet change that you're gonna get. It obviously looks terrible here, looks really great on the poster, looks even better on your computer screen. And if you really want, you can come talk to me about the new product that we're just finishing up right now, the development of, which is a gridded ice mass change product. So this is a height change product, right? We can calculate volume from that. We're doing all of the Fern corrections for you now. And so you can just pull off the internet a very nice gridded ice sheet mass change product for whatever your needs for mass change are. It's gonna be really great. It's gonna make getting mass change at kind of the kilometer level incredibly easy for everyone here. So look forward to that. Come chat with me at the poster.
Hi everyone, I'm Tasha Snow and I'm a glaciologist and oceanographer at NASA Goddard and ESSEC, University of Maryland. And my poster is about CryoCloud. If you haven't seen CryoCloud, it's a state-of-the-art cloud computing platform and community that's been funded by the Transform to Open Science program and ISAT-2 Project Science Office at NASA. And it's a cloud computing platform that is meant to provide us with easy access to the cloud so that we don't have to build our own infrastructure. And we're doing a lot of training in how to streamline your workflows. There's an example of that on the right. There's a lot of computing practices that we're learning how to do much better in this new technological world. And we're providing Python environments, R, MATLAB, those kinds of things you can all use in CryoCloud. And we have over 600 users now. So if you want to learn more about how to improve your computing, streamline it so you don't have to download, so that it's faster and that you store a lot less data, come talk to me at the poster. And I can also tell you a lot more about the cloud.
Hello, my name is Paul Summers. I'm a postdoc at Georgia Tech and Rutgers University. My poster is a bit better formatted, so if you want to come and learn about how melange, ocean, and glacier processes all interact with each other, which is mystically hidden behind these black boxes, you can come find me on my poster and learn all about it.
Hi, my name is McKenna Zemak, and I'm a PhD student at Texas A&M Corpus Christi, working with Lindsay Prothrow. And so I'm collaborating on a project looking at Antarctic ice sheet extent and dynamics in Pinell Basin in the Ross Sea. So in progress of that, I looked at 3 cores, looking at the diatom record, physical properties, and grain size. And so this is just one of my cores. And so what we see is a really complex sub-ice shelf setting with varying assemblages of reworked diatoms. So if you would like to know what a reworked diatom is, or see the rest of my cores, please come by and see my poster. Thank you.
Hi everyone, my name's Hannah Verbancourt. I'm a 4th-year PhD student at Colorado School of Mines, and my poster is all about determining the characteristics of basal ice and subglacial conditions under a coupled ice stream pinning point system. And we do that through unsupervised clustering methods, where we essentially come up with a regional cluster of all the different characteristics under the ice stream and pinning point system. And we're hoping to connect that to some sort of buttressing feedback that we're seeing in these systems. So, thank you.
Hello, I'm William Weber. I'm a graduate student at LSU. I've had the pleasure of working under Dr. Philip Bart, and the purpose of my poster is to present preliminary results on an ongoing investigation of the stratigraphic evolution of Ross Bank. And so using a recently acquired grid of seismic data, we've been able to locate several grounding line proximal features and interpret a system of aggradation. And so the purpose is to find when differential erosion, on the onset of differential erosion, and understand how and when Ross Bank became a pinning point. So looking forward to meeting all of you. Thanks.
Thank you. So those are all the official posters. We now have a bonus slide from Ellie and Ben. I'll let them explain.
Hi, I'm Ellie Abrams, and I'm a data science postdoc at Stanford.
I'm Ben Hills. I'm a postdoc at Colorado School of Mines.
And we're here to advertise a workshop happening just after WAIS. So if your flight is not leaving right away, come stay. We're going to have the first WAIS workshop proceedings. So if you're presenting at all at the conference, either a poster or a talk, please participate in our proceedings. You can upload a PDF, either of your poster or your slides. But you can also come learn how to make a Jupyter book, which is a nice interactive online publication in which you can share code, figures, write up a description of any of these things. So it's a nice way to supplement anything that you've presented here. And when you submit it to our proceedings, in exchange, we will give you a DOI. So this is something that you will be able to share on your CV, and it becomes citable. So if maybe you developed some kind of code routine to pre-process data or to create some cool visualization, this is a great place to share that, make it available to the WAIS community, and make it citable so that you can get credit when people actually use your tutorial later. So,
I'll just add that if folks can't stay on Thursday, if you have like an early afternoon flight, then we'll send out an email and people can still contribute to this. We're going to keep it open until January, so yeah, no pressure to stay on Thursday if you can't.
Great, thank you. So now is an opportunity for any new WAIS attendees to briefly introduce themselves and let everyone know what kind of research they're interested in. So thank you.
Good morning, everyone. My name is Jordan Tucker. I'm a first-year master's student at Stanford University working with Dusty Schroeder. And what gets me excited, I don't have a poster this year, but what gets me excited is sea ice, any and everything to do with sea ice. So if you love talking about sea ice, I'm your guy. And really how extratropical cyclones affect this sea ice, that's kind of what I'm looking at right now. I'm very much in the early stages, but I'm super excited to get started, and I'd love to talk with anyone. Thank you.
Good morning, everybody. My name is Franco Migliolo. I am a environmental engineering master's student at University of Florida, so I can tell you that the gators are real. And I shared with Jordan that there's a video of a horse fighting a gator, so you guys should check that out. It's pretty interesting. So environmental engineering is kind of a broad topic, but I like it because it's multidisciplinary. I worked in the field for 7 years, and prior to that, I was a chemist. Prior to that, I was a biologist. So expect me to ask you a lot of questions because I'm very interested in a lot of things. And there's one thing in life that I haven't done yet, and you can imagine why I'm here, for the Antarctica part. So it's one of my future goals is to be part of, work with, collaborate with something related to Antarctica. So this brings me here, and I expect to be here next year too. Thank you.
Hi everyone, my name is Lindsay Manito. I'm a third-year PhD student here at the University of Florida, working with Rob Hatfield. So I come from the paleo perspective, working with marine sediments, and I'm actually a Greenland person here, eavesdropping on Antarctic conversations, which I think based on this morning's discussion, we can see has some interesting connections. So I'm excited to talk about any and all things, proxies, age models, marine sediment perspectives, and Northern Hemisphere and Southern Hemisphere connections. Thank you.
Hello, my name is Nathan Shadle, and I'm working at UF at the Gator Glaciology Lab under MacKie. I'm a lab technician there, and I also work during my undergraduate at the Gator Glaciology Lab. I work on accelerating interpolation algorithms for mapping subglacial topography, and we have a Python library called GStatSim that's soon to get an update where those algorithms will be parallelized, so look out for that. I also have worked on projects for looking at diffusion models and generative adversarial networks and seeing how they're able to reproduce this spatial statistics of an ensemble of geostatistical simulations. And yeah, I'm very excited to be here. Thank you.
Hi everyone, my name is Jason Dreber. I'm a second-year PhD student at Colorado School of Mines, working with Dr. Ryan Venturelli. I don't have a poster here, but I am interested in applying cosmogenic nuclides to glacial processes, glacial systems. I did a lot of undergrad work looking at Laurentide retreat, and right now I'm doing a lot of method development for in situ radiocarbon. And trying to develop new, or trying to expand which minerals we can use so we can use cosmogenic nuclides in different parts of Antarctica. So talk to me about that if you're interested.
Hello, I'm Isabel Cordero. I'm not new to WAIS, but I'm new to I recognize many of your names from submitting data sets to us, so it's really nice to see some faces to go with those. I'm one of the 3 curators, and I'll be here on Thursday afternoon to talk about data access and distribution and submission. So if you have any questions throughout the conference because you're not available on Thursday afternoon, please come up to me and chat at any point. I'd be happy to talk about it. So yeah, nice to meet you all. Thank you.
Hi, I'm Orion Schomber. I am a master's student with Brad Rosenheim at the University of South Florida College of Marine Science, finishing up my master's, woo. I work with 3 different fern cores on Larsen C, and I am looking at the cyclical nature of the isotopic signatures on them, and seeing if those signatures align with various climatic modes of variability.
Hi everyone, my name is Sunmi Zhao. I'm a first-year PhD student at Stanford working with Dusty Schroeder, and I'm interested in the question of how bed topography impacts subglacial hydrology, and also thinking about some of the paleo records and what that can tell us about subglacial hydrology, and just looking forward to learning from everybody here.
Great, thank you so much to everyone for introducing themselves, and can we please thank our poster presenters again, everyone who's spoken. Thank you. And yeah, I encourage you to please take the time to go to the poster session later, which is at 4:30. Is that 4? Later. Go check them out. Thank you, everyone.